All right, so if we're looking at the details here of this second messenger biological signal transduction pathway, all right, membrane receptors and these associated proteins are going to do one of two things. So here you can see we have our signal molecule binding to that membrane receptor on the outside or on the plasma membrane. All right, remember it can't go through the membrane, so it has to bind to that receptor on the membrane. That binding to that membrane receptor will initiate the next response. Now remember, this is your first messenger. This is going to initiate this conversion, remember this signal transduction. Remember we have this transducer that will initiate another set of sequences. All right, and so typically it can do one of a couple of things. It can activate protein kinases that are found in the cytosol. And remember that those protein kinases were enzymes that are able to transfer that phosphate group from ATP, all right, to protein, which was the protein that phosphorylates other enzymes within the extracellular fluid. All right. The other option is it can activate an amplifier enzymes, or like an amplifier sequence. All right, and this is what's going to create your second messengers. All right, and in a couple of ways that it does it, it can alter gating of ion channels. All right, so you can see this right here where it alters the gate of an ion channel. The other option is it can increase the intracellular calcium. And when you increase intracellular calcium, you will activate these calcium binding proteins. All right, and then you'll have this metabolic response. Finally, the other option is it can change the enzyme activity. All right, like the protein kinases or these phosphorylated proteins that remove and add these phosphate groups, and it can lead to this conformational change of a protein to create the response. And we've kind of already been over that sequence before. So those are your options for these signal transduction pathways in biological systems.